So far, we've spent a bunch of time and energy talking about the fundamental building blocks of living things, so all about atoms, molecules, and polymers, and now we're going to jump up 2 levels of organization to talk about cells. We're going to start out this section with a bit of a history lesson. I need to add a little disclaimer that at the moment this history lesson is going to be very white, male, and Eurocentric. And although this is not the only way to introduce the study of cells, for what it's worth, it will include a couple of interesting stories that we'll encounter along the way. So we're going to start this story back in the 1600s, which is a period characterized in Europe by being a seafaring colonial era for people such as the Dutch, the French, the Spanish, and the English. And during the colonial era, people from these cultures were very busy traveling to other parts of the globe to collect anything and everything of value that they could find, return to Europe, and then hopefully sell those things for a nice profit. And part of what this meant is that among the upper class members of European society, natural history became a very popular hobby because biological specimens were things that could be bought, sold, and traded. This was also a period during which there were some technological advances in glass lens production. So think about the production of telescopes to explore space. And some of these advances in glass lens production also meant that people could engage in some more local exploration of tiny things at home. And while these seafaring exploration and the technological advances were going on, always remember that culturally there's often a lot of things happening simultaneously in a given culture. So there was also a question that was very hotly debated in European society, and that question is, how do living things arise? And in other words, how do living things come into existence? And this is a question that comes up in, I think, pretty much any culture. But at the time in Europe, there was a particular hypothesis that had gained some footing for explaining just where living things had come from. And that hypothesis, as I've noted here, is called spontaneous generation. And spontaneous generation actually had its origins in the thinking of Aristotle. And the idea was that living creatures could arise spontaneously from non-living matter, and that this is a very common way for new creatures to come about, with specific organisms coming from specific substances. So as one example, people thought that fleas could arise spontaneously from dirt and dust, and they thought maggots could arise from meat that was left lying around. And Aristotle even had a really sophisticated explanation for how different kinds of clams and scallops each came from different kinds of ocean substrates. So, sandy ocean, rocky ocean, etc. So anyway, all that said and done, people weren't entirely clear about, for example, where exactly babies came from, but that was basically seen as some sort of extension from the origins of simpler creatures from simpler substances. And along with this general idea, people did also have the notion that there is something else required, some kind of vital substance found in the air. So that's kind of symbolized here with this candle. So that was the state of thinking at the time. And it may have only been a matter of time before someone came along with a very clever way to test this spontaneous generation hypothesis. And the guy who gets credit for this is an Italian dude who had doctoral degrees in both medicine and philosophy by the age of 21. So his name is Dr. Francesco Redi. And Dr. Redi seems to be the kind of person who's really good at careful observations. So a lot of his earliest observations actually had to do with figuring out how snake venom actually works. And he may have been the person who first came up with the whole idea of having a control treatment in an experiment. So in any case, Dr. Redi was kind of skeptical of this idea of spontaneous generation, and so he decided to go about testing it. And the experiments that he came up with involved, one of these examples, the origins of maggots in meat. And so what Dr. Redi did is he came up with 3 different treatments for his experiment. So he had one set of jars that contained some meat that were left open to the air, as you see here. And then he had a second set of jars that contained some meat that were sealed off with corks. This is kind of just capped here. So totally sealed off so nothing could get in or out. And then he had a third set of jars that were covered with a very fine mesh cloth. So those jars could let the vital substance in, but they were still separated from the surrounding environment. Okay, this is a good point to take a moment and pause the video, because what I want you to do is to say what you would predict for Dr. Reddy's experiment if this theory of spontaneous generation is in fact correct. What would your predictions be? And then we'll take a look at his actual results. Okay, so now here are his actual results. Okay, so on the meat in the open jars, he found maggots, and he even found some flies. Okay, on the meat in the closed jars, he found no maggots. And then on the meat from the jars covered in cloth, well, he didn't find any maggots on the meat, but he did actually find maggots on top of the mesh covering the jar. Honest results. So here's an example of where a very clever experiment got used to strike a blow against a very popular hypothesis. So, what do you think Dr. Reddy's conclusion is here? Was the spontaneous generation hypothesis supported? I think not. All right, so at the same time that Dr. Reddy was conducting his meat jar experiments, there are also a number of other things going on, all that stuff I mentioned with telescopes. And so in addition to pointing lenses up at the sky, other people also started to point lenses at tiny things and then started to draw pictures of what they were seeing. I kind of think of this as being like, the TikTok or Instagram of the 1600s. People are totally hooked on this tiny universe that couldn't be seen. So one of the guys who was getting really popular for his drawings of tiny things was this guy, Robert Hooke, who is also an architect. We don't have any pictures of what he looks like, so we just have to imagine, that he's a pretty cool dude. So he was a guy who one day happened to stare at some pieces of cork and saw that the cork had a very uniform appearance consisting of a bunch of tiny chambers. So here's a drawing that he created of that. And he thought those tiny chambers looked like the cells where monks lived, so he coined the term cells. And then he went on to write this best-selling book about his discoveries, Micrographia, and so those ideas got popularized through his work. And at the same time, there's a second guy, pictured here, named Antony van Leeuwenhoek, who had started to tinker around with this microscope technology. And in his case, he was actually initially really interested in seeing the cloth fibers for his drapery business more clearly. But over the course of that effort, he managed to make some incredibly good lenses using some secret methods that he took with him to his grave. But then he was able to use his lenses to directly observe living cells. And Van Leeuwenhoek never published any books, but he did write a ton of letters to the Royal Society of London about his observations. And one of the things that Van Leeuwenhoek observed was living cells as they're moving around, which led him to the idea that cells come from other cells. But this idea, so Robert Hooke agreed with him, okay, seems like that's the case. Even so, this idea was not universally accepted. So we have to fast forward to the mid-1850s to where things stood with thinking about cells. And collectively, this set of ideas are referred to as cell theory. And there are a whole bunch of guys who get credit for coming up with this list. So first component of cell theory relates to all living things, the idea that all living organisms are composed of one or more cells. Second tenet of cell theory is that the cell is the most basic unit of life. And now the third tenet here is that all cells arise only from pre-existing cells. And now this last idea was actually still controversial in the mid-1850s. Although it had been shown that spontaneous generation didn't happen for larger organisms, many people still considered spontaneous generation to be possible for cells. I mean, after all, why not? So Then again, in 1860, so a good 200 years after Van Leeuwenhoek's observations, a scientist came along who developed an extremely clever set of experiments to once again smack down this new microscopic version of the hypothesis of spontaneous generation. And this new guy was a Frenchman by the name of Louis Pasteur. So the big challenge was, how could he still provide this mysterious substance in the air that seemed to be very necessary, and yet demonstrate that cells will not grow unless there are already cells present? So Pasteur's innovation was all about flasks that he designed. He created this set of flasks that had these very long, thin, curved, and tapered necks. And here's a photo of some of Pasteur's actual flasks that are still in the Pasteur Museum in France. So you can see this very long, thin, tapered neck. And so what that allowed him to do was the following. So he filled these flasks up with a nutrient-rich broth, and then he boiled the broth. So he's also the guy who's responsible for showing that if you heat a liquid above a certain temperature, you can use that to kill the microbes in that liquid. And so that's where we get the term pasteurization. So then, after boiling the broth to kill off the previous microbes, he would then let the flask sit for a while. And if microbes started to grow again in the broth, then the broth would start to turn cloudy. But when he did this with one of his flasks, the flask that just sat there just did not turn cloudy. It sat there for a long time. So, okay, that's kind of boring. But, there are always doubters. So, people wanted to know, well, could it be something about this whole process that is making the broth so it's no longer nutritious, and that's why there aren't microbes growing? So to persuade all the doubters, he repeated this experiment, boiled the flask, let it sit, but this time he cracked the neck of the flask after letting it sit. And shortly after he cracked that neck, bam, he had cloudy broth. So bacteria showed up. And he also had a third treatment group where he tilted the broth in the flask and sent the broth into the neck. And there again, the result was cloudy broth. So that helped to demonstrate that it wasn't anything about the flask design itself that was preventing microbial growth. So Pasteur was able to really say, well, take that, doubters. And so once again, a very clever experiment helps to eliminate the spontaneous generation hypothesis for cellular origins. So that now brings us to the modern version of cell theory. The first 3 points are the same, but there's now a fourth point added. So this fourth point is that cells contain the hereditary information of the organisms of which they are a part, and this information is passed from parent cell to daughter cell. And the amazing thing about this last point is that it means that there is unbroken continuity between the modern cells in the world around us today and those very first primitive cells that appeared on Earth. And that is pretty amazing. And I've alluded to a little bit of this earlier, but there have continued to be some incredible advances to microscopy since the time of Van Leeuwenhoek's microscope in the mid-1600s. And those advances have continued to contribute a lot to our understanding of cells. So I'm going to talk for a minute about 3 main types of microscope images, plus a couple of other tricks. And you're sure to encounter a range of types of microscopy over time. So the 3 main types that I'm going to highlight right now are light microscopes, transmission electron microscopes, and scanning electron microscopes. And without getting too far into the weeds, the way these work is light microscopes simply shine a light through a thin slide, and then we view the light that passes through the object of interest with our eyeballs. And this microscopy method can usually get us up to a magnification of around 1,000 times what you can see with your eye by itself. The next type here, the transmission electron microscope, is a similar idea, except instead of passing a light through a sample, electrons get passed through a very thin sample suspended on a grid. And then this gets detected on a screen. And the advantage here is that electrons have a smaller wavelength than visible light, so it's possible to get clear images of even smaller objects with a transmission electron microscope compared to a light microscope. The third type of microscope I've highlighted here is a scanning electron microscope. We actually have one of these up in the Saint Center here in Morel. So for this method, samples are often coated with a thin layer of conductive metal, and then electrons are shot at the surface of the sample, and then the signal bounces off of the sample and gets detected by a detector. So scanning electron microscopes, or SEMs, are really great for imaging the surface of an object in great detail. So now here's some examples of some microscope images using these three technologies. So up top we've got rabbit sperm cells visualized using a light microscope. And then in the middle we've got a single sperm cell visualized using a transmission electron microscope. So you'll notice it's a very thin cross-section. We can see a lot more detail. And then on the bottom here we've got those same sperm cells visualized with a scanning electron microscope. So you can see all of the surface character of these cells. Okay, and it hasn't stopped there. So by now scientists employ all kinds of other tricks, such as using specialized stains to highlight certain features, using different kinds of lighting, which will bring different types of structures into focus. They'll use things like fluorescent dyes, so some of that's pictured down here. And it's also possible to use fluorescent antibodies to mark specific structures. And then there's a whole host of computerized methods that can be used to sharpen images. And so now we can do things like this lower image here has a bunch of vesicles, a cellular structure that have been tagged inside of a living neuron. And you can actually watch these vesicles move in real time down some of the branching structures in the neuron. And so collectively, these tools have all expanded our ability to make observations at different scales. And collectively, those many, many observations have contributed to the generation of the hypothesis about how all living things are related to each other. And so here's a picture of that hypothesis again from the start of the semester. And this all now brings us to a point where we're prepared to start looking at some cells. And we're going to be exploring both prokaryotic and eukaryotic cells. And I'll talk a little bit more about how we're going to do that in the next video.